Hello everyone and welcome. I'm the host of today's meeting, Jessica Tu, from GenScript Biomarketing Department. On behalf of GenScript Bio, I'd like to thank you all for attending today's webinar. GenScript Bio, a subsidiary of GenScript Biotech Corporation, is a global player dedicated to providing premier end-to-end service from discovery to commercialization with professional solutions and efficient processes to accelerate drug development for customers. Today's webinar will include 2 talks. Each talk will take 30-minute presentation and 15-minute Q&A session. You can submit questions at any point during the webinar using the chat or Q&A panel situated in the menu on the right. Now, I'm delighted to introduce today's first speaker, Dr. Li Chen, the Senior Director of Biologics Discovery Department of GenScript Bio. After obtaining his doctorate in immunology from the University of Massachusetts School of Medicine, he joined GenScript in 2015 and has been responsible for antibody drug discovery platform for the past eight years. He has more than 200 project experience in antibody drug discovery and engineering innovation optimization, as well as 3 patents related to biopharmaceutical discovery. During today's webinar, Dr. Chen will be discussing utilization of VHH, phage display library and integrated protein engineering platform to generate antibody leads for CAR T therapy. Now, I'm delighted to hand over to today's first speaker, Dr. Chen.
Thank you, host. Thank you everyone for taking the time to join this webinar. In the first session, I will, as the host introduced, I will talk about the antibody discovery strategies for CAR T therapy through the VHH phage library and antibody engineering approach. We will start with a quick introduction of the challenges and the considerations in CAR lead discovery, followed by a detailed discussion on corresponding solutions. In the last part, to help everyone better understand the workflow, we will share case studies of a VHH lead generation for CAR T application against 2 targets, the CD7 and the RR1. Currently, CAR T cell therapy is approved for the treatment of B-cell relapse or refractory leukemia, lymphoma, and multiple myeloma. It has led to excellent response rates depending on the patient population and therapy. Despite promising efficacy, CAR T cell therapies is associated with significant side effects. The 2 most notable toxicities are cytokine release syndromes and immune effector cell-associated neurotoxicity syndromes. Overall, a careful risk and benefit balance exists between the efficacy and the toxicities of the therapies. The challenge lies in the physiology and the physiology of a CAR T-related toxicity upon the activation of a CAR T cells. In general, these are the typical limitations and potential strategies. For the CAR T cell associated toxicities, people commonly alter the CAR structure to alleviate the toxicities. In addition, cytokine antagonists and a CAR off switch like rituximab can also serve as alternative remedies. For on-target off-tumor effect, it is crucial to select appropriate targets. Affinity and avidity modulations of the antibody can also contribute to the differentiated TAA recognitions. For antigen escape, it is a new trending to target multiple antigens, such as bispecific CARs. For the immunosuppressive microenvironment, combination therapies with immune checkpoint inhibitors offers alternative solutions. And for the CAR T cell trafficking and the tumor infiltration, local administrations like the intratumoral injections is being investigated. A typical CAR T design includes four domains. The extracellular binding domains delivers the target specificity through the binding between the antibody and the tumor-associated antigens, aka TAA. Those antibody key features like the affinities, avidities, aggregations, epitope locations plays a crucial role in CAR T cell specificities and the potencies. The spacer domains provides flexibilities to the extracellular binding domains and leads to further impact on the efficacies and toxicities. Transmembrane domains in CAR structure serves as a fulcrum to transducing ligand recognition signal to the intracellular cytoplasmic domains, while the cytoplasmic domains usually contains T cell activation CD3 domains and multiple co-stimulatory domains to provide sustained signaling and to promote T cell proliferation. The typical workflow of CAR lead discovery starts from target identification and antibody screenings. After selection of antibody, these candidates will be built into the T cell by proper design and virus delivery, followed with cell-based functional characterization and efficacy studies in animal models before testing on human. Next, let's move on to the solution part for the CAR lead discovery. In the lead generation stage, several technologies, including hybridomas, single B cells, human naive libraries, VHH immunized libraries, and the VHH naive libraries are available at GenScript Bio. These approaches will generate CAR leads in either single chain FV or VHH formats. Single chain FV used to a mainstream option since it can be easily converted from IgG. However, this conversion usually leads to affinity loss, sometimes by the range of 10 to 100 fold, not to mention the risk of aggregation. Alternatively, VHH is a naturally generated antibodies with retained affinities. In addition, the size of the VHH gives it a unique advantage for certain inaccessible epitopes. In this talk, we will focus on the VHH modalities through the immunized and naive library. Typically, antibody from immunized animals tends to have a better affinity, but also the risk of immune tolerance. And it also takes longer time for the whole process. Alternatively, naive library is much faster, but at the cost of a potential low affinity. In addition to the source of the library, the antibody formats also plays a crucial role in lead generation. Typically, the antibody fragment is fused with the phage through the P3 protein. Such a big chunk attached to the antibody may interfere with the binding kinetics to antigens, leading to false positive or false negative in the screening assays using the phage supernatant. With the design of an amber stop codon here between the antibody and the P3 protein, the phage constructs can be induced to express the antibody in to the supernatant in a soluble versions, which will significantly reduce the non-specific binding noise during the ELISA and FACS screening, and also facilitate the high throughput affinity rankings by SPR bureau hardware. Here I'm listing all 5 different approaches for car lead generations for a side-by-side comparison, including timelines, pros, and cons. Hybridoma is definitely the most cost-efficient ways to generate the antibody leads, but it takes longer, while single B cell is relatively faster with slightly higher cost. Naive library, both the human and the VHH naive library, tends to give better diversity and a shorter timeline, but it may need affinity maturations with extra costs and timeline. Immunized library is still the most popular strategy for VHH lead discovery in the CAR T context. In the lead optimization stage, antibody humanization is a common practice to reduce the immunogenicity risk, while affinity maturation is only required for antibodies with relatively low affinities, such as those generated from the naive library. Currently, the humanization work takes less than 3 weeks at GenScript Global, including the sequence design, the humanized candidate purification, and affinity measurement by Beerport. Besides humanization itself, a panel of developability indicators such as post-translational modification hotspots, immunogenicities, and aggregation propensities can also be included during this stage. In this humanization case studies, we designed 4 humanized variants based on the wild type sequence, and 3 out of these humanized sequence demonstrated almost identical affinity kinetics as the wild type. If the lead antibody is not optimal in terms of affinity, in vitro antibody maturation by random mutation within the CDR regions is a common strategy to improve the affinities. Considering the number of possible combinations of a total random mutation with all 60 residues, it will be impossible to conduct continuous random mutation across the whole CDR regions. Alternatively, we implement 3 approaches to keep the mutation library in a manageable scale. The first step is to narrow down the mutation sites by structure modeling. MOE software is commonly used for homology modeling to identify CDR critical residues that contribute the most to the antigen antibody bindings. Then the single residue saturation mutation library will be created for each of these CDR critical residues. In this library, all 18 amino acids except the cysteine and the self amino acids will be evenly distributed by the technology called precise mutagenesis. Once the beneficial single residue mutation are identified by SPR affinity rankings, they will be incorporated into a combinatorial library to screen for the best combination of the mutations contributing most to the affinities. Once the lead sequence is finalized, we will move forward to the next stage, the in vitro functional validation. Typically, 4 indicators will be taken into considerations during the cell-based functionalities. The CAR expression level on primary T cells, especially adequate percentage of CAR positive cells, are crucial for T cell activation. Cytotoxicity is the primary measurement of effector cell activation as determined by LDH release from the lyse cells or the luciferase from reporter cell. Cytokine release assay is another key approach to demonstrate effector cell activation through interferon gamma, TNF, and interleukins. In addition, effector cell proliferation can be determined by the CFSE staining. The general workflow of a CAR-T functional in vitro functional validation starts from vector design, followed by T cell transfection, expansion, and end up with functional assays. The whole process takes about 2 months. Primary cell-based screening is one of the most popular approach to evaluate CAR functionalities. Here I'm showing 2 case studies, one for the CAR-T and one for the CAR-NK. Both CARs expression were confirmed by flow cytometry, first followed by cytotoxicity evaluation on different ET ratios or in a real-time killing kinetics. And the induction of interferon gamma is also monitored with both case studies. Sometimes primary cell could be a little bit tricky to handle. Alternatively, reporter cell-based screening can be a more robust way, especially when you need to screen through dozens of candidates. In this system, an IL-2 promoter-driven luciferase genes is incorporated into the Jurkin cell, and the potency of the CAR T cell can be determined by either target cell lysis rates in the PBMC on the top panel or the activation window upon the co-culture with target cell as shown in the bottom panel. The last panel in the whole process is the in vivo pharmacology. It includes efficacy, PKs, and toxicity studies. The typical animal model studies last about 4 to 5 weeks. During this course, we will measure the tumor size, the body weight for efficacy, the circulating CAR T cells for effector cell proliferations, and the amount of effector cells infiltrated into the tumor tissues. These 4 are the most common assays we will conduct during the in vivo animal model stage. Next, to help everyone better understand the general process of a car-lead discovery, I will share two internal case studies. The first case study is CD7 VHH discoveries. CD7 is a marker for T-cell lymphoma and leukemia. Most of the CAR T therapy targets B-cell malignancies by T-cell cytotoxicities. However, in order to kill T-cell cancer with T-cell, it is crucial to make sure that the CAR T-cell will not kill itself through the CD7 markers. There are three approaches to address the self-antigen-driven killings. Either the CD7 gene knockout by CRISPRs or the blocking of the cell surface expression of the CD7 by ER retention, or the sorting for CD7 negative T cells as the source of CAR T. In this case study, we will be using the ER retention strategies. This is the candidate screening funnels. We started with alpaca immunizations by recombinant proteins. After several rounds of panning and screening with protein and Jeter cell, we obtained 31 unique sequences for purification with FC fusions. The top 15 candidates were selected for further affinity characterizations. Then the top 3 out of the 15 binders will carry out with humanizations and CAR T functional validations. During the library screening stage, we conducted the SPR affinity ranking with the soluble supernatant. Most of these candidates demonstrated an off rate around 10 to the -5 to 10 to the -7, which is considered as a pretty good affinities for antibodies, for therapeutic antibodies. In addition, ELISA binding affinities and epitope competition assays were carried out with the benchmark antibody, the PA3-17s. All of these binders showed similar affinities and the same binding epitopes as the benchmark. After reviewing the affinity data and the sequence cluster, top 3 candidates were selected for humanizations. All of them showed affinity at sub-nanomolar range before and after the humanizations, at least 10-fold better than the benchmark, the PA3-17. Following humanizations, these 3 candidates were constructed as a bivalent VHH on the CAR Ts. As I mentioned at the very beginning, we utilize the ER retention approach to eliminate the CD3 expression on the CAR T cell to avoid the self-killings. The CD7 cell surface expression level was closely monitored after the transfection, after the CAR T transfections. It reaches the peak on day 5 and gradually comes down to background levels post the transfection. Comparing to the benchmark antibody, the P3-17, one of the top candidates, the AHP 19485, demonstrated better retention of the cell surface CD7 markers as 98% versus 84% for the CD7 low T cell. In addition to the retention of the CD7 marker, the expression level of the anti-CD7 VHH CAR was also monitored by flow cytometry. All top 3 candidates and the benchmark showed 90% plus expression rate. After expression confirmations, luciferase-based reporter cell killing assays was conducted on 2 different target cells, the Jurkat cells and the CCRF cells, to evaluate the in vitro efficacies. In these cytotoxicity assays, the CAR-T candidates showed similar potencies as the benchmark at different ET ratio. In the second study, it is a VHH CAR discovery against RR1. RR1 is a typical tumor-associated antigen, or called TAA. It is upregulated in most B-cell malignancies and solid tumors, but with limited expression in healthy tissue. In recent years, it is one of the most popular drug targets for a variety of therapies, including CAR-Ts, ADCs, T-cell engager, bispecific antibodies, and the monoclonal antibody drug. The main challenge of this campaign is that RR1 shared extremely high homology among different species, including human, monkeys, rodents, and alpacas. It is unlikely to induce immune response in these animals by immunizations due to the immune tolerance. To tackle with this challenge, instead of alpaca immunizations, we utilize the naive library approach for the antibody candidate identifications. After pairing with recombinant RR1 protein containing different extracellular domains and screening against both protein and overexpression cell lines, 30 unique sequences were recovered for purifications with Fc fusions. Then the top 23 candidates were selected for further affinity characterizations, followed by CAR-T functional validations with 8 selected candidates based on the sequence cluster. Finally, 3 top binders were carried out with humanizations and affinity maturation. Unlike the immunized library as I shown in the first case studies, the naive library generated a panel of binders with differentiated profiles, including diversified binding affinities and epitopes on different domains on the extracellular part of RR1. Based on the affinity profile and the sequence cluster, 8 selected binders were constructed as a monovalent VHH on the CAR-T. The expression level of anti-RR1 VHH CAR was monitored by flow cytometry. All of them show roughly 80% expression rates on the primary T cells. This is the primary cell, not Jagger cell. After expression confirmations, luciferase-based report cell killing assays and cytokine release assays on IL-2 and interferon gamma were conducted to evaluate the in vitro efficacies. In these studies, 3 candidates showed comparable potencies as the clinical benchmark, the R12. These 3 top candidates were further humanized, and the affinity was successfully retained at the 10 to the minus 7 level. And as we discussed earlier, the antibody recovered from naive library tended to have a relatively lower affinities. So one of the humanized candidates, the AHP 15485, were selected for affinity maturations. The affinity is improved by 30-fold, reaching the single-digit nanomolar range. Before I end this session, I would like to give a few words about ourselves. GenScript Bio is a reputable biological discovery solution provider with speed, quality, and cost efficiency. As a professional CRO plus CDMO, we have 20 years of experience in therapeutic antibody discovery, and so far we have successfully delivered more than 1,800 discovery and engineering projects, among which at least 16 pipelines are in global clinical phase, and 2 products are already launched in the market. Our offers of target to PCC services with diversified platforms and flexible business models aims to accelerate our partner therapeutic drug development. And that's it. Thank you everyone for your attention. Let's move on to the Q&A session.
Thanks for Dr. Chen's excellent presentation. If you have any questions, please type it on the chat or Q&A panel. As you enter the questions, please also follow our LinkedIn account, GenScript Bio, and the Korean audience can also scan this QR code and follow our Kakao channel. There will be more online sharing resources, and we will share them on our LinkedIn and Kakao channel at the first time. Thank you. Okay, we received some questions from the audience. Let's take a look at the first question. What is the difference between the VHH vs IgG in terms of humanization?
Yes, so basically IgG contains 2 chains, the heavy chain and the light chain. So when you do the humanization, you need to consider both chains. While for VHH, it only contains 1 heavy chain. So the overall process is a little bit simplified. However, there is a little bit tricky for the VHH immunizations, although both VHH and IgG adopt the same strategy of immunization, the CDR grafting plus back mutations, because in the VHH framework, there are several conserved residues that need to be considered during the framework replacement and back mutations. Other than that, as I mentioned, the general strategy of humanization for both modalities are pretty much similar.
Okay, thank you for your answer. And next question is, what are your selection criteria of the antibody candidates?
For CAR Ts, it's a little bit different from therapeutic antibodies. For therapeutic monoclonal antibodies or bispecific antibodies, the key indicators is, of course, the functionalities at the earlier stage, especially whether it can induce or inhibit the signaling pathway. However, for CAR Ts, uh, it technically is challenging to conduct function assays at the very beginning until you reach the stage to conduct to build the CAR Ts and to validate the cytotoxicities in cell-based assays. So during the antibody screening stage, usually what we pay attention to is either the binding affinities. It's not that the binding affinity is the higher, the better. Usually we will intentionally select binders with different coverage of affinity range from 10 to the -8 or even -7 all the way to 10 to the -11, because you never know whether the one with lower affinity or the one with higher affinities can actually achieve a better balance between the potency and the safeties. So that's the first criteria, a broader range of affinities. The second indicator is the sequence. It also aims for better diversities. So usually these are the two criteria we uh, uh, we uh, we put a lot of weight on it to during the early stage selection.
Okay, thank you for your sharing. The next question is, do you think VHH is a better option comparing to SCFV when it comes to building block of CAR domain?
Well, as I mentioned in the discussions, SDFA definitely has its own advantages. You can easily convert a IgG into a single chain FV so that you do not have to spend a lot of extra resource and time to generate a new candidates. Especially, you can you can design a car with a sequence from an expired patent. However, if you look at the therapy itself, VHH definitely offers several advantages, including the better affinities, less work of engineering, and less risk in developabilities, especially the aggregations that leads to the tonic signal.
Okay, yeah, thank you. That's all the questions I received by now. If no more questions, let's move to the second topic.